So good afternoon, everyone, again. I think we can start this session. I would like to firstly thank our speaker, Dr. Thomas Winkelmüller, and I hope that's how it pronunciates, who will present this session on high-quality RNA isolation for qPCR and RNA sequencing, which was one of the topics that researchers from CIMER highlighted as one of the most needed in their training needs. So I would like also to thank the Neoscience company in the person of Mr. Faisal Al-Bahar, whom we have contacted. So briefly, Dr. Thomas Winkelmüller manages since 2020 the international sales for the Macherey-Nagel Bioanalysis portfolio in parts of Europe, the Middle East and Southeast Asia. During his PhD at the Max Planck Institute for Plant Breeding Research, he focused on the evolution of transcriptome responses during immunity in various plant species. So I would like to thank him again for his presence in our seminars. And I would ask him to please start his presentation, for which he will have around 45 minutes, followed by some 15 minutes for questions.
Perfect. Yeah, so I hope you can hear me.
Yes.
So thanks, Katerina, for the very nice introduction, and thanks a lot for the great opportunity today to give that presentation to you.
Thank you.
Yeah, thanks as well, Neoscience, for giving that really nice opportunity. So as Katerina already introduced very nicely, my name is Thomas. I'm working for the company called Macherey-Nagel. And I will just give you a brief introduction into the next 1 or 2 minutes. And then we will jump right into the topic about how we can help you to isolate the best and highest possible quality RNA for your downstream applications. Good. So first of all, as I know that many people in UAE or the Middle East are not that aware about a company, I will give you a very, very brief introduction about Macherey-Nagel. So basically, what our mission at Macherey-Nagel is to make your nucleic acid purification as easy, fast, affordable, and reliable as possible, and that for a broad variety of different starting materials. So no matter if you work with clinical samples, with environmental samples, microbiology samples, plants, and so on. So we have nucleic acid purification for all these kind of samples. So Macherey-Nagel is really focused on quality. So we are a Germany-based company. So all our production R&D is based in the western part of Germany. We have all kind of quality systems in place to make sure we can supply the highest possible quality products. And we have a sort of trusting our solutions worldwide. We have a lot of different research publications of researchers citing our product, and we are very proud that we can help these researchers to achieve their everyday goals in the lab. As well, I would like to mention that we have a very highly skilled team back in Germany. So we have many of us with PhD degrees in genetics, molecular biology. So whenever you have troubles or have a question concerning isolation of DNA and RNA, you're highly welcome to contact us. And of course, as well, our distributor Neoscience here in the UAE has a very, very strong team with a lot of professionals in molecular biology, so they can also help you every time. So yeah, that's just the picture of our company. So actually, my office is usually located in this brickstone building here. And as you can see, we have a mixture of old and new. So our company is about 100 years old, but we are constantly expanding and growing. And that's always by focusing the customer benefits. So all we can do is really centered to give you the best experience. Okay, so with that really short introduction about Macherey-Nagel and what we are doing, I would like to jump into the topic, and this will be our agenda for the next, I would say about 40 minutes, probably. So first, I would like to give a bit of introduction about, a bit of RNA isolation in general, why it's important to isolate RNA, why it's important to have high quality RNA, but also what are the a few of the typical challenges that you need to solve when you work with RNA. Then I would like to take a chance and show you some of the solutions that Macherey-Nagel can offer to help you in the lab. So I would first of all tell you something about our nucleospin products, which are the mini spin kits, which are very nice for the manual isolation of RNA. Then we will take a look at our, there's actually a typo here. That should be nucleomag RNA. So it's a magnetic bead-based kit for RNA isolation, which can be nicely automated. And after that, we will take a closer look to some of the kits we have specifically for clinical samples, as I think some of you might be interested in that as well. So how to isolate total RNA from FFPE samples and other clinical samples as well. Okay, let's start with the introduction. So when we talk about RNA, what do we actually, what are we actually talking about? So there's actually not one type of RNA, but there are multiple types of RNA, as probably most of you know. So when people talk about RNA isolation, for most of the times, they're looking for the mRNA that codes for proteins to really analyze how the expression of the RNA is connected to the protein expression in the end. But as well, we have several other RNA species, like the ribosomal RNA, which actually makes up most of the RNA. So if you typically isolate RNA, most of the stuff you will get is ribosomal RNA. And we also have some more specific RNAs, like the microRNAs, which also play a very important role in cell signaling, but are, in contrast to mRNA or ribosomal RNA, very, very small. So this is actually an important thing. So before you start your research in your product and you're searching for the right kit, you need to think of what kind of RNA you actually would like to isolate. Because for different types of RNA, there are also different solutions and different kits that can help you. So yeah, I mean, why do people look for RNA? Why it's important to isolate RNA? I mean, simply RNA has a really, really important in the signaling of the cells. So RNA regulates many, many biological pathways, and thereby it's really important to understand what kind of RNA molecules are expressed in a specific state. For example, if you compare disease cells to control cells and so on. And if you want to quantify RNA, really, you need to, first of all, isolate all molecules. So the product or the kit that you use needs to make sure you get all the RNA that you need to do the analysis. You need to isolate intact molecules, so you don't want to have fragmented RNA. You really want to have high integrity, long stretches of RNA. And of course, that's also a very important point that the isolation system needs to ensure is that you don't have any inhibitors in your isolation. So you have nothing that inhibits your PCR enzyme or the enzymes you use for your library preparation and so on, because otherwise you will get issues to nicely quantify your RNA and expression. Okay, so what makes RNA a bit special compared to DNA isolation, for example? So it's quite simple. So DNA is pretty stable. So if you have an isolated DNA, you can leave it on your benchtop for room temperature, and most of the time it will be very fine. In contrast, the RNA is especially sensitive to degradation. So this is, of course, because of its structure. It's not a double helix, singular helix, and it's very sensitive to RNases. So to the nucleases that can cut the RNA into small pieces. And the bad thing about these RNases is they're all around. You know, they're on your hands, on your clothes, just everywhere on your bench. So it's really important, or really important to consider right from the start when you take your sample and you want to isolate RNA. You need to think of how do I really protect my RNA molecule to not get degraded RNA? So just two very simple ways to do that is, first of all, using shock freezing. So if you have really cold temperatures, if you put your samples into liquid nitrogen, that's one of the most efficient ways to, first of all, make sure you have a high RNA quality, but also make sure you stop all the processes that are going on in your cells. You know, cells can change the expression really rapidly. So if you treat them with some treatment or something, within seconds, you will have a massive reprogramming of the transcription. And therefore, it's really crucial to really stop and take a picture of what you really want to look at. Another way to do that is with chaotropic salts. So the chaotropic salts will inhibit the enzymes, the RNases, that will cut the RNA. So this is really important. And this is a substance that we usually include in all our lysis buffers from our kits. So as soon as you have your sample homogenized within the lysis buffer, you can be sure that the chaotropic salts are protecting your RNA from further degradation. And that's also one of the reasons you can do the procedure with our kits on room temperature, and which is quite handy and comfortable. So what do you as researcher typically look for if you're searching for an RNA isolation product? So first of all, as we just said, you would like to get an RNA of high quality and high integrity, so long stretches, non-fragmented RNA. To solve that, we include the chaotropic salts in our buffers. Then, of course, also important for many of you, you would like to get rid of your DNA. So you would like to get an efficient genomic DNA removal. So there are many ways to do that, and two ways that we included in our products is either the kits will come with the rDNase enzyme to do the enzymatic removal of DNA, or they will come with the gDNA removal column. So it's a very convenient column that's very quick to remove your DNA. Of course, you want to have a fast kit. You would like to get your RNA as fast as possible, so we make sure you have a quick processing at room temperature. And of course, we always keep an eye when we design our products that you have or get the highest possible yields from your samples. Okay, so now we already talked a lot about the quality of RNA, but I would like also to take a look, how do we actually measure the quality, and how do we make sure the RNA is really good for my downstream application like the RNAseq or the qPCR. And there's basically two major ways to do that. So one thing that many people do is they measure spectrophotometrically the amount of RNA they have, and also they check for contamination. So they check how the RNA absorbs at 260 nanometers of wavelength, and you get a specific value. Many people do that with a nanodrop or similar instruments. And importantly, and that's what many customers come to us, they say, oh, I'm not sure about the quality of my RNA, and I have this and this nanodrop measures. You need to be aware of that. The nanodrop tells you something about if you have a contamination in your sample, so for example, the PCR contamination, and it tells you a bit of if the RNA is present or not. It doesn't necessarily tell you anything really about the quality of the RNA in your sample, because even if the RNA is highly fragmented, you will get decent spectrophotometrically measurements. Therefore, we have another measurement. It's called the RNA integrity number that you can typically do on the bioanalyzer. So there you really measure what you try to detect are the major subunits of the ribosomal RNA, and you can calculate a ratio of these, which is the so-called RNA integrity number. And the closer that is to 10, the nicer is your sample. So usually if you're working with cell cultures or with tissue, and you take the sample very freshly and you make sure no RNA is degraded, you can get values around 9 or 8 to 10, so very high. If you have some environmental samples, a bit different. So usually for plant samples, it's around 7, 8 if you have a good quality. So that really ensures you have long stretches of RNA that you can send for your sequencing or for your qPCR. Okay. With this being said, I think we leave the introduction here, and I would like to give you or present to you some of the solutions that Macherey-Nagel can offer for you to isolate high quality RNA. And so first of all, I would like to introduce to you our nucleospin RNA Plus kit. So this is a really nice kit if you do manual, or if you want to perform manual RNA isolation. So it works with our nucleospin columns, so that are usually mini spin columns you can process in a benchtop centrifuge. And the advantage that you have by using the kit is it's ultra fast, so it includes a gDNA removal column. That means you don't need to wait to 15 minutes for the DNase enzyme, but it's just a quick spin. It gives you very high purities and RNA integrity numbers. One thing that's I think is particularly nice, which many other kits on the market do not have, is we don't need any reducing agent for that kit. So you don't need any stinky beta-mercaptanol, beta-mercaptanol, which is actually a very nasty chemical in the lab. So you don't need to work on the fume hood, just on the bench, everything is fine, and you are safe and protected. Of course, it will give you high yield, and also very nice feature is we have an XS kit that we will take a look at in a second. So to sum it up, you really get a quick and easy product and a really clean, high quality RNA. So this is just a procedure. So for those of you who ever used a spin based DNA or RNA isolation kit, it should be familiar to you. You lyse your tissue or cells. Then you have the first step, which is filtration to really make sure that in the end your binding column does not clog. And as well, within that step, you have the removal of genomic DNA. Then you bind your RNA, you have several wash steps, and you elute a very pure, highly pure RNA. So that in total is a very fast process. So here we just have some application data where we compared the time we need to process a sample with our kit compared to some other kits on the market. And there's also one of our kits, so that was a bit older generation that many people use because they're happy with the kit. So that has an enzymatic digestion, which you see here in gray, which can take up a quite large amount of time. So now if you get rid of that, you can really isolate high quality RNA within 20 minutes. So I think that's very nice, and that's really also faster than most other competition on the market. Yeah, so here we have some data and some proof that also we can get RNA of very high integrity values. So here you can just see one example of mouse spleen, where we usually get an average win value of 9.4. So that's really great quality. It's ready to send that for library preparation for your RNAseq. And here also we have some other data where we compared our kit to the very popular QIAGEN RNAseq kit. And you can really see that the removal of genomic DNA is on a similar level. So we basically did a qPCR for DNA. You can still detect it because PCR is a really sensitive method. So you will never be completely free of DNA. That's actually, if you go into depth and you really search for it, you can always find 1 or 2 molecules, but you really are on a good level compared to the market leading quality here. Yeah, then one other thing I would like to introduce, which I think it's very interesting for all those of you who have troubles because their sample sizes are too small. So either if you do cell sorting and you only have a small amount of cells, or if you have a very tiny tissue samples or biopsy samples, for example, then we have these XS version here. So it's also called nucleospin RNA plus XS version. So the plus indicates that also the same technology with the gDNA removal column. But now we have a bit different silica spin column, which has kind of funnel, which you can nicely see here. So that's XS version on the left side. On the right side, you have the standard version. So you funnel down to really small area of the silica membrane, and by that, what you can achieve is you get a very small elution volume, and you really concentrate down your sample a lot. So here's just some data to really prove that we will be able to go down to even individual cells, and we could still detect the RNA that we could isolate it from these individual cells. So I think it's really good if you have very small sample amount. You should definitely give it a try here. Just to show you some examples that a lot of different groups and researchers around the world are using our kit successfully. I just have some examples here of some very recent, very nice papers published in Nature Biotechnology here and in Nature. So the one of them using our RNA plus XS kit, for example, for their cells. And yeah, so both of them actually use the plus XS kit. And also, which is an interesting thing for you, is that these kits, for example, work very well together with the kits that, for example, Takara offers for the downstream library preparation, for example, or the enzymes Takara offers. That's also something that our partner Neoscience UAE can provide to you. So you have really a complete workflow when you use a Macherey-Nagel kit next to Takara products. They work really great together. Yeah. Good. So just losing very few words. This is actually the bit older version, so you don't see the plus. So it's a nucleospin RNA kit. I would say you could say it's oldie, but it's a goldie. So it doesn't have that gDNA removal column, but the advantage you have with that kit is on the market for a long time, for over 20 years now. And the advantage you get by this is you have many different methods. You can use it for a variety of different starting materials and so on, because people and researchers around the world tried whatever stuff using that kit. So it has a very long list of support protocols for different applications, and you can just come back to Neoscience or us and we can help you with that. It includes the enzyme, which is not the case for any of the competition products. So you have everything included that you need. And interesting for you, that's something that's not available yet for our Nucleus Plus kit, but here, so for some of you that might look to isolate DNA and RNA simultaneously, we have a buffer set where you can also elute your DNA, so you get both DNA and RNA in a simultaneous isolation. This is also available in the XS format, yeah. And importantly, it really supports many different applications. One other thing is, now we only talked about the spin column and XS column, but we also have many other formats. We have a MIDI column, so this column here, which if you have a larger sample amount or you just need more yield, this is a good kit for you. Of course, we also have these kits available for higher throughput. So for medium throughput, we have these 8-well strips, so where you can process a multitude of 8 samples, either using a centrifuge or using a vacuum. And of course, we also have these 96 well plates where you can process 96 samples at once, again with vacuum or centrifugation. And this can also be automated on any kind of open access liquid handling platform. And of course, we support you. We're happy to support you with implementing that automation if you look for that. Good. Then I would like to get to the next kit we have. So now here, I don't have a typo from the agenda. So it's a NucleoMag RNA kit. So it's a magnetic bead-based kit. As you see up here, I mean, it looks like just a brownish kind of liquid. So that's how actually these magnetic beads look like. And I would like to show you how that works in just a second. So the main advantages by using a NucleoMag kit is that you're really flexible. So you can do it manually, just doing 5 or 10 samples, but you can also easily scale that up, both manually or automated. So you can do 96 well samples at once and a 96 well plate using 8-channel pipette, for example, or you can automate that on various platforms. And also you can adjust nicely to the sample amount you would like to use. So by just changing the amount of beads that you use, you can either scale down, take less beads, and then you can do more samples with the kit, or you scale up if you need a lot of RNA. And this is this kind of illustration here just gives you an example how the kit is processed with a magnetic watt system, like for example the Kingfisher Flex. With these systems, it's quite nice because what you have is you transfer the magnetic beads that the nucleic acid bind to. So first of all, you have a binding step. So you see the greenish and orange RNA and DNA molecules here. They will bind to the beads. You can then remove the beads from the liquid and thereby getting rid of all different types of contaminations. Then you can do the digestion of DNAs if that's needed for you. Then you erase orange molecules here. You are left with the RNA. You have 2 or 3 other wash steps, depending on the nucleomag kit. And this really makes sure you have very pure, very clean RNA in the end, and then you can elute that and you're ready to go. This is a very fast procedure by just transferring the beads to the different wash buffers and other buffers. You can process 96 wells within 20 minutes if you have such a magnetic watch system. So this is a very nice way to automate and increase your throughput. Here I also brought some of the application data that we created with the kit, so really to show you that it works really great with different samples. So here now we have some data from human cells and from tissue samples, where you can see that you get a really great yield from around, I would say 20 microgram to up to 40 microgram, of course, depending on your sample type. And most importantly, also indicated by the orange dots here, you get very nice CT values in the qPCR. And this really shows you that you not only get high yields, also get high quality RNA that can be amplified, and you don't get any qPCR inhibition in here. This is also some bioanalyzer profile from the RNA isolated from cells, and you can see that the Win values are indicated here. And as I told you in the beginning, 10 is kind of the best you can get, and as long as you get somewhere between 8 and 10, it really shows that you have RNA of the highest quality, and you can be sure that you have really great results in RNAseq. That was actually on kind of human samples and cell samples. Maybe some of you also work on other parts like environmental samples. So also would like to show you that the kit works very well for that. So here we have some application data, plant samples, so soybean, wheat, maize, Arabidopsis, tobacco, which is usually have much more PCR inhibitors, but still you can see that you can get very good PCR results. And also here you see some examples for bioanalyzer profiles from wheat, from maize, from tobacco, Arabidopsis. So usually you always have, depending a bit on the sample, Win values at least above 7. So as I said, usually plant samples always have a bit lower Win value, but these are very, very good for sending them out and do your downstream sequencing. Good. Now I would like to come to a few of the kits, or the more specialized kits that we have for different clinical samples. First of all, I would like to start with the kit that we have for FFPE samples, so formalin fixed paraffin embedded samples, which is a particularly challenging and difficult sample type. And those of you who work with the samples probably know what I'm talking about. It's a great sample type because the samples are conserved for many, many years, so you can keep them 20 years and you still have the sample conserved. But the downside of that is that you usually have a very poor quality of nucleic acids, so both DNA and RNA. They're often degraded, and the amount or the sample amount that you can use is usually quite limited, so you have very small input amount of samples. So we really try to make a kit to address many of these challenges. So first of all, you need to get rid of all the paraffin that you have in your FFPE samples. So many other kits on the market use xylene for that, which is actually a quite nasty toxic chemical. So we developed a new buffer, which is xylene-free and non-toxic. So we have a patent on that buffer system. So it's really nice for you, as you don't need to work with any toxic substances. Then, very importantly, we have a very efficient D-cross-linking technology. So as the name says, formalin fixed. So basically, your DNA is fixed and connected. I mean, it's illustrated here by that all these DNA molecules stick to each other, but they also stick to other proteins and other contaminants and stuff. So you need to make sure to remove all these cross-links, because otherwise you cannot amplify the DNA in your downstream application, or also your RNA. So the D-cross-linking step really makes sure that your nucleic acid strands are separated from each other and accessible. This total RNA kit can isolate, as the name says, the complete RNA, so not only the large RNA molecules, but also your microRNA molecules. And that's important because most of the time the RNA is degraded in these samples. We also have this available in the X version. If you said we have, again, very less starting material, you can go down to very small elution volumes. And we also have a magnetic bead-based kit to isolate the RNA from the FFPE samples. So again, showing you some of the application data. So we compared our kit in blue here to different other kits that are on the market. And we really took a look if we can get both the messenger RNA as well as the microRNA. And you can see by the lower CT value here, so a lower CT cycle always means you have a better qPCR performance, that we have the best qPCR performance here. And again, that's true for the microRNA sample. So you can see that also some kits really fail to isolate small, very small microRNA fragments. So that's the thing we make really sure when you use our kit. Again, we have many publications with that, and you may ask, okay, if my DNA or if my RNA is that degraded and I've not enough RNA there, can I actually do an RNAseq? And the nice news here is that, again, our partner Takara has very nice kits for that. Also, if you have a low input amount or highly degraded RNA, still it's worthwhile looking at the kits for the library prep, as they have specific kits adapted to that. And this is also used by a lot of researchers around the world. So here you can see they use the total RNA FFPE-XS kit from us together with RNA sequencing kit from Takara Bio. Good. With that, I would like to move. So we stay a bit similar to what we just discussed, so about total RNA isolation. So as I said in the beginning, you should always think about what you want to do with your RNA. So do you simply want to measure the expression of messenger RNA? Then you will find with most of the standard kits. But if you're looking for smaller RNA like microRNA, you need to make sure that the kit's adapted to that and also is able to bind these very small nucleotides of around 15 to 20 nucleotides in length. So this you can do by using our nucleospin microRNA kit. Although the kit is called microRNA, it will isolate total RNA. So it will isolate both the small RNA as well as the large RNA. And you can basically choose if you would like to have that in one fraction, or if you would like to have a fraction with the small RNA and another fraction with a large RNA. So yeah, it's a bit illustrated here. It seems a bit complex. You could also precipitate your proteins from the same sample if that's of interest for you. But then, first of all, you really bind the large RNA and the small RNAs in flow through. You can isolate then the large RNA, and with a flow through, you bind to another column, and then you, in the end, can isolate both small RNA and large RNA. Good. Besides of that, we don't use any phenylchloroform, which is used in many kits to isolate microRNA. So again, you don't have any toxic substances. You can conveniently work at the bench. We have a nucleospin filter included, so you make sure your binding column is not clogged, and also it helps the homogenization of the samples. And of course, we have the DNase enzyme included as well, so you can as well make sure that you get rid of DNA. But again, you can also omit that step if you say we want everything, we want total RNA, we want protein, we want DNA. So this would be a great option kit for you, as then you can isolate the DNA as well. Good. Let me take a sip here. Yeah, so this is again some application data we have for nucleospin microRNA kit. The picture quality is not the greatest, but I think you can see what I would like to say. So if you want to isolate total RNA, so you have everything in one fraction, you get here the peaks for the ribosomal RNA, so the very large RNA fragments, and you also get a small peak here for the microRNA. So if you would separate the fractions, you would see in a bioanalyzer only the small RNA peak. And importantly, if you would analyze that with a bioanalyzer, you need a specific bioanalyzer kit for that. Otherwise, it will just tell you your RNA is bad. You don't have RNA in there, because usually the bioanalyzer looks for these 2 ribosomal RNA peaks. That again you would find them in the fraction of your large RNA. So to sum that up, it's a really efficient isolation of all types of RNAs, and you can include either DNA and protein as well. Good. Then the last part of the talk is about the other kits that we have, clinical RNA. So I won't go too much in detail here, but I just would like to give you an impression what other products we have. We have a really large product portfolio. So today we only concentrated on the RNA kits, but of course we have all the kits for DNA, from environmental samples and so on. Good. So of course, I would say one of the most typical samples used in clinical research is of course blood. So for sure we have a very nice kit for RNA isolation for blood samples called nucleospin RNA blood. That's also available in a MIDI format. So if you have large amounts of blood, that would be a great kit to go for. And yeah, we many times get also the questions from customers, how can I actually automate my RNA isolation for blood? And there we also have the very nice 96 well spin kit for you that you either can do manually with H on the pipette, you will be much faster, or you can also use a liquid handling station that has a vacuum manifold included, and you can fully automate the isolation here. You also see some of the application data where we compared our kit in the blood in a blue color here to the QIAGEN kit, and you see that for both samples that we looked at, we get superior qPCR values, which again shows we can efficiently rid of PCR inhibitors that you have in blood. So hemoglobin is a very potent inhibitor of PCR, so you need to make sure that you get rid of that. Then we also have a dedicated kit to isolate microRNA from plasma samples. Here you again have a really simple, fast procedure. You don't have any toxic compounds involved. Yeah, it's also, here we have a nice solution if you look for microRNA that's associated to exosomes, which is often the case. We have an exosome mutation solution that can use for the kit, and then you really can either concentrate the microRNA in front of your isolation process. And again, some application data here, where in blue we have the qPCR data from our kit, and we compared that to some of the competitors, and we really get superior qPCR data for these microRNA that you can find in your plasma samples. Maybe importantly, the reason why we have a dedicated kit for plasma, because we talked about the microRNA kit for cells and tissue, is plasma is very rich in proteins, and here you need to make sure that you get rid of all the proteins, as there would also be an inhibition afterwards. So we really have a specialized washing industry on that. Good. Then we have also RNA kit for stool samples. So these are again also very, very good samples. Stool has a lot of humic acids and other potent PCR inhibitors. So this is also a kit including some of our B tubes, so it really makes sure you have a mechanical lysis. You lyse a lot of microbes in the stool, again, you get a really good and great qPCR values, so you have a very efficient removal of PCR inhibitors and superior PCR quality. Good. As I just mentioned to you, we have several other RNA kits as well, for example, for plants, or for soil and other stuff. And on our homepage, we have that very nice RNA purification guide, which gives you a very good overview about all different kinds of RNA kits that we have. And yeah, if you just look for the right kit for you, I can encourage you to take a look at that. And also, as I said, you can always feel free to contact us. So with this, I would like to thank you for your attention. I think, yeah, we took about 40 minutes, so we're quite good in time. I hope you learned a lot of things. And yeah, we are really happy to support and help you. If you have any questions about RNA purification, please go ahead, contact Neoscience, our partner here in UAE, or contact us directly for technical questions. And yeah, we are more than happy to help you.
Thank you so much, Dr. Thomas Winkelmüller, for this very interesting presentation. I open the floor now to questions.
I see at least there's some questions we have from the chat here, when I see correctly. So maybe we can go with that. So is it possible to use nucleus spin to isolate bacterial or viral RNA from stool? Yeah, it is. I mean, that's basically the kit, one of the kits I showed in the last slide. So our nucleus spin RNA stool kit. Yeah, basically, I mean, the RNA molecules, of course, from bacteria or from viruses are no different than the RNA molecules we have in our cells. So as long as you can make sure that you have a strong lysis buffer chemistry so that you can homogenize and lyse all the bacteria and viruses in your sample, you will also get the RNA from these bugs. And yeah, as I mentioned, our nucleus spin RNA stool kit includes these, we call them bead tubes. So they have small glass beads that you use in the first lysis step, and these really make sure you also efficiently lyse gram-positive bacteria, for example. So this is definitely a kit you can use. Yeah. Then there may be another question. Do we need special kit for RNA extraction from sputum samples? So here again, it's a bit the question what you would like to do. So if you would like to isolate the host RNA from the sputum samples, or if you would like to look for pathogenic RNA in your sputum samples. So if you want to do the latter, looking for pathogenic or viral RNA from the sputum samples, we have specific kits for that. So I didn't cover that in the talk here, but we have, for example, our nucleus spin virus kit, which many groups around the world also used for the COVID-19 PCR testing to isolate the viral RNA from swabs, and that also works very well for sputum samples. This is really, the kit is designed to be highly sensitive. So even if you have very small amounts of the pathogenic RNA, the kit makes sure you can get that out and amplify that. If you rather are looking for host DNA, you can also just try simply to use our standard nucleus spin RNA and nucleus spin RNA plus kit, which I think that is a bit more orientated to getting you a really high yield. Yeah, and that would also be a good choice and should work. Yeah. Good. Perfect. Yeah. Any other questions or?
Yes, I have a question. If possible. Hello?
Yeah, I can hear you. Yeah.
Yeah, I have a question. Thanks for this interesting presentation. I've used your kits before, and they're good kits. Back to stool RNA extraction, you said something about including beads. Do these beads come with the kit, or I have to do that separately? And number two, if I use the beads, is it enough to use vortexing in the stool sample, or do I need to use a special bead beater?
Yeah, very good question. So your first question, do the beads come together with a kit? Yes, they do. So you will get everything in the package, and the bead tubes will be within the kit. And the second question is, do I need a vortex or homogenizer? You can basically do it on the vortex. That works pretty good in our hands. Actually, it's important that you don't vortex the samples vertically like you would usually do, but you need to put them horizontally on the vortex. So we also have for the Vortex Genie 2, which many labs use, we have an adapter. Yeah, it's difficult to tell, but it's kind of a blue styropor adapter that you can put on the vortex, where you can put multiple tubes inside, so that you can vortex, I think, about 10 samples at once. But vortex is possible as well. But if you have a homogenizer, like a wedge system or fast prep, Priscillis, that's also possible, totally possible. Yeah.
What do you recommend for storage temperature once you isolate your RNA? It will be in the elution buffer now. Now this elution buffer is what? It's probably just depth sea water or what?
Yeah, so it's either, I think it depends a bit on the kit, but it's either very low salt buffer, or it's simply RNase-free water. So RNase-free water would usually use to elute your samples. So that's a very good question, because once you elute your samples, they're not protected anymore. So I would always recommend, if you eluted your samples, put them on ice directly. So for example, if you want to quantify your RNA with a nanodrop, put them on ice, do the quantification, and then freeze them at -80. If you know that you need the the RNA for many downstream applications, it's also always a good idea to do aliquots before, because what you wanna try to reduce is freeze thawing samples. So it's not good to freeze and thaw your samples for, let's say, 5 times or more. So that's kind of the good practice, and -80 usually make sure that your RNA is not degraded.
Okay, can I ask something like a stupid question? How does the kit how does the kit compare to something like triazole?
Triazole, I think it's a bit unfair comparison. I think for most kind of common cell or samples like common cells or tissues, you will get a very similar yield, so that's comparable. Triazole has several advantages. It's pretty aggressive. Those of you who use it probably know it. It's a pretty nasty thing. You need to go in the hood. You need to protect yourself. You don't want to be splashed with phenol and stuff. So that's a downside, of course, of triazole. So working with our kids, it's much more convenient. But of course, what's true is that a triazole can help with very, very difficult samples. So very difficult to lyse samples, for example, or specific plant species. That's why also we have dedicated kits to specialized samples, because you cannot simply not do all the sample with just the one kit. So if you work with plants or with very hard to lyse samples, we have specialized kits for that. So always feel free to contact us. We always have support protocols as well for hard to lyse tissues. We also have an analog to triazole. It's called nucleosole. So it's kind of our own version, which can be good if you just have many samples and use for a cheap solution. That can be used as well. But I think the major downside is really that it's really nasty and toxic. But
I'm thinking in line of triazole, in line of like in stool samples or similar ones. Something like triazole is really tough on these inhibitors, these substances, like a humic acid, things in blood or things in stool. Do you really think that your stool extraction kit for RNA, it will really get rid of all these inhibitors in something like stool? Or do I need to use the triazole with the other combination?
You can totally feel free to try that out, the kit. Our hands, we developed that knowing that we have that much humic acid and inhibitor substances in the stool samples. That's the major point at designing a specialized kit for that. Otherwise any kit could do. We really worked hard to get the lysis chemistry and everything, and the wash buffers that are so stringent that you get the clean sample. So usually there's no issue for qPCR inhibition or any inhibition for RNAseq. So
Thanks.
You're welcome. Good. Yeah, I don't know if there's no more questions.
If nobody is asking, can I ask an extra question? Can you overload the column?
Yes, you can. Definitely you can, yeah. So overloading, I would say it's one of the most typical errors that customers do, because many customers think, you know, the more, the better. But that's not necessarily true, because what happens is you can overload basically you can overload like the lysis chemistry. So I mean the lysis buffer usually that is specified for a specific amount of sample that we specify in a user manual. And yeah, sometimes you can go a bit higher, but at some point there's just not enough lysis buffer for all the cells. So you don't lyse enough cells anymore, so you have a lot of unlysed cells. And then what happens is that your yield really drops. So it's a misconception that increasing the sample amount is always increasing the yield. So sometimes even you get better results with less sample amounts, especially if we talk about samples with a lot of inhibitors. So if you, for example, take way too much a stool sample as a start, even though if you might get a bit more yield, you probably get more inhibitors as well, which in the end, it doesn't help you because you cannot use the RNA that you isolated. So I would really stick to the recommendations that we use we have in the user manuals. Yeah.
Comparing the MAG and the nucleus spin, I mean toe to toe or head to head. If I use the same starting material of my sample, which one will give me more yield?
I would say it's a difficult question. I would say there's no major difference, and there's no black and white answer to that. So probably there's a sample where you get a 5% more using the beads, and maybe there's another one you get 5% more using the silicon membrane. But you know what usually happens is both the beads as well as the silicon membrane have a specific binding capacity. So let's say they can bind 100 microgram of RNA. So usually you never really reach that capacity, because that's theoretically, if you would put pure RNA on there, it can bind all of that. But what happens is in the binding step, the silicon membrane will also bind all kind of other stuff that you don't want to have, like proteins, the PCR inhibitors, and other stuff. And then you use the wash buffers to get rid of that. So you never really reach that total theoretically binding capacity. And therefore, it's also, I think, quite difficult to say which one gives you better yield. I mean, I would say there's no major difference that it's only half with one of the other technologies. So with both, you get good yields and then very high quality. I mean, one thing that's, of course, different with the beads, as I said, are scalable. So if you say, I need more yield, you can, of course, use double amount of beads, double amount of washing buffers, of lysis buffers, and so on, and you will likely get double amount of yield. Or vice versa, if you say, I always get 10 microgram, but I only need 5 microgram, you can scale down. You take half, and in the end, you save costs on your reagents. So yeah. I hope that answers your question a bit.
I have a question. Do you guys produce any other kits other than RNA extraction?
Yeah, we do all kind of nucleic acid isolation extraction. So what we do not have is, you know, let's say cloning enzymes or NGS kits and stuff. So as I mentioned with that, of course, our distributor Neoscience has all that in the program. So you can always contact them to help you with that. For example, the Takara range, which is very great for RNAseq, that we don't have. But for nucleic acid purification, not only for RNA, but also for DNA, we have a pretty complete set. So also for your DNA isolation, high molecular weight DNA, DNA from blood, DNA from plants, so whatever you can think of, we may get you covered eventually.
Thank you. Do you guys have any PCR or restriction enzymes?
No. So we as a company, Macherey-Nagel, we do not have that. So that would be on Neoscience. So yeah, you please feel free to contact our distributor Neoscience. So they have all kind of restriction enzymes and stuff like that in their portfolio. Yeah. Good. So if there's no more questions in the interest of time. Yeah,
we are finishing our time, unfortunately. If there aren't any more questions, I would like to thank again Dr. Thomas for the wonderful presentation. Thank you so much for your time. And anyone who wants to reach you, it would be easier through your emails and the company. Thank you so much again, and thank you all for your presence in this CIMER seminar.
Thanks a lot.
You.
Present with a really great pleasure, and thanks for all of you who attended. And yeah, have a nice afternoon and evening.
You too. Thank you so much.
Bye.
Thank you so much.